Can we genetically engineer microorganisms to be smart to manufacture more effective therapeutics like cannabinoids? My name is Nick, and I'm a second-year bioengineering PhD student in the Ian Wheeldon Lab. I'm working with CRISPR tools and molecular sensors to produce high-purity cannabinoids like CBD and yeast. Of the over 100 cannabinoids in the cannabis sativa plant, only a handful have been well studied in humans. And this is really a shame because despite these compounds being very similar in structure, they have incredibly different effects on human receptors. Like, take a look at CBD and CBDV on this receptor called TRAPA1. The difference between these two molecules is barely noticeable to our eyes, but it turns out our bodies know the difference. Now, we can get massive quantities of these cannabinoids from hemp plants, but they're all mixed together. Rare cannabinoids, meaning not CBD or THC, usually only account for less than 1% of the total cannabinoids in the plant, and trying to chemically separate something like CBDV from CBD is incredibly difficult. In 2019, researchers at Berkeley tried to remedy this problem by taking some genes from the cannabis plant and incorporating them into yeast. Turns out the enzymes not only produced only small amounts of things like CBD and THC, but they produced a mix of compounds just like the plants. So this is the challenge that I'm trying to tackle. I'm using CRISPR-targeted genome-wide mutagenesis and mutant PYR1 sensors to artificially evolve yeast that make more and more pure cannabinoids. So let's break that down a bit. CRISPR is a revolutionary gene editing technology that allows us to delete or insert genes very precisely. The system uses a protein called Cas9 that acts like a pair of scissors to cut DNA. Importantly, these molecular scissors are guided by the appropriately named guide RNA. After the cut is made, the cell attempts to repair the break, but usually isn't very good at it and ends up resulting in a gene knockout. Now, what if we had a guide RNA for every single gene in the genome? This would allow us to knock out every yeast gene one by one to make a mutant library of organisms, some of which may produce more and more pure cannabinoids. So we have a library of these mutants, but we need a good way to sort through them. So let's just make the yeast smart and let them sort themselves. PYR1 is a hormone receptor naturally found in plants. When this hormone is present, the protein changes shape ever so slightly so that it can bind to another protein called HAB1. If we can attach PYR1 and HAB1 to two other proteins, one that binds to DNA and one that tells the cell to read the DNA, then when the hormone is present, the cell reads the DNA, a process called transcription. Researchers at UC Riverside have found that this PYR1 sensor can be mutated to be sensitive to new compounds. Here, a mutant sensor was put in plants in order to warn the plants of a drought. Smart plants! In my research, I've developed PYR1 sensors that are sensitive to cannabinoids, of course, and they can tell the difference between very similar structures, just like the receptors in our bodies. So, my main project in my PhD program is to first create a strain of yeast that makes some amount of CBD, then incorporate a cannabinoid PYR1 sensor to make the yeast know what cannabinoid they're producing, and finally, I can use CRISPR genome-wide mutagenesis to knock out every gene in the genome one by one, and the sensor will allow me to sort for mutations that result in more pure and higher amounts of CBD or any other cannabinoid. So, metabolic engineering of microorganisms allows us to make powerful therapeutics more efficiently and in greener ways. More efficient production of these chemicals means that costs come down for researchers and consumers, allowing for more affordable and better studied treatments for you. Hey, thanks for watching. I'd like to thank SciComm for hosting this event. Science communication has been something that's been really, really important to me for a while now. And so seeing events like this is really fantastic. I'd also like to thank all the sponsors listed on the screen here. And with that, thank you so much for watching. Bye.